Hello, my name is Mansoor Diop. I am a fourth year at the University of Minnesota Rochester. Along with my group mates, we will be presenting bacteriophages found in Rochester waters. We are health science majors here at the university. Bacteriophages are viruses that specifically target bacteria. They insert their genome inside the bacteria. The bacteria then uptakes this genome into its own genome. It is then transcribed and translated back to bacteria. The phage proteins then reassemble inside the bacteria and it's able to burst through the bacteria. Bacteria can be found in waterways from fecal contamination. The analysis of phage can determine the presence of these fecal-borne bacteria in waterways.
Hi, my name is Leah Tucker. I'm a third year at the University of Minnesota Rochester, and I'll be covering our methods. We got our samples from Silver Lake and Zembro River, and so we got soil samples and water samples. For soil samples, we initially diluted it before doing zero dilutions. For the water sample, we just went straight into the zero dilutions. We then added 3 drops of chloroform and then centrifuged it for 5 minutes. And then we then transferred the supernaut from that mixture, from that centrifuge into a sterile microcentrifuge tube using a filter that only allows the bacteriophage to flow through, and added our E. coli broth and let it incubate for also 5 minutes. We then mixed that mixture into a soft agar and pour them on top of plates and let them incubate for 24 hours.
My name is Jessica, and I'll be talking about the result. In the first photo, we have an agar plate that has E. coli and bacteriophage in it. The small circles that you see is where the bacteriophage had killed the E. coli, showing that there is some phage in it. To confirm it, we isolated using phage lysate. To clear the water, it shows that there is phage in it. So the middle tube and the right tube shows that there is some phage inside. And the image on the right shows the bacteriophage capsid captured at the Cornell Research Institute.
My name is Yasin, and I'll be covering the conclusion and future directions portion of our project. So in conclusion, we were successfully able to isolate bacteriophage from Silver Lake water samples. The soil samples in our analysis, the soil samples yielded more phage than the water samples. So the Silver Lake soil samples showed bacteriophage, whereas the water samples necessarily didn't. The Zumbro River water samples and soil samples were all inconclusive, and so we couldn't really say whether or not we found bacteriophage present. But as for our future directions, we will continue to assess that water source and try to see if we can find any phage there as well. Additionally, we will share our refined phage extraction procedure with a new student group in hopes that they can proceed with it for future use. And lastly, we will assess bacteriophage quantities in Rochester waterways. The little QR code in the corner shows our list of references that we used for this presentation. Thank you.